So now we are going to study about purine synthesis. Actually, it is of 14 steps and quite difficult to remember either for your board exams or your for your NEET PG entrance. So I'll just simplify it and we'll concentrate on where you have to focus a lot. All right. So purine synthesis, it requires two things. First thing it requires is a lot of energy, that is in the form of ATP. It requires a lot of ATP. We'll see how that is. And second is it requires a substrate. That substrate is ribose 5-phosphate. The substrate is ribose 5-phosphate. So where is this ribose 5-phosphate coming from? This ribose 5-phosphate is coming from HMP shunt pathway. Remember HMP shunt pathway, oxidative phase, non-oxidative phase. Yeah. So we get ribose 5-phosphate from HMP shunt. Energy, where do we get this energy from? We get this energy mostly from glycolysis as well as the TCA cycle. Mostly it is the glycolysis and HMP shunt. Mostly it is a glycolysis and HMP shunt. Why I'm saying this is because in RBCs, suppose if you're talking about RBC, red blood cell, remember there is no mitochondria, so no TCA cycle occurs. Only two pathways will occur. The glucose 6-phosphate that is there in RBC has only two things to do. One, it can undergo glycolysis. The second, it can undergo HMP shunt. This has been very beautifully explained by the role of band 3. Role of band 3. How band 3 or anion exchange protein 1, how it will regulate glycolysis and HMP shunt inside the RBCs. It has been beautifully explained. Perhaps in some other class we'll talk about differential regulation of glycolysis by band 3. All right, for now just remember that the energy that is coming from is from glycolysis and the principal substrate is ribose 5-phosphate. All right, so we have two things. One is ribose 5-phosphate and a lot of energy or ATP. So what happens to this ribose 5-phosphate? Ribose 5-phosphate. So there is a ribose. So this is a ribose C1, C2, C3, C4 and C5. Okay. So this ribose, to this ribose on the carbon 5 there is a phosphate group which is attached. Ribose 5-phosphate. Okay, this ribose 5-phosphate is converted to a pyrophosphate. That is, a ATP molecule is utilized. An ATP molecule is utilized and it forms AMP. So two phosphate groups from ATP are taken and they are given to ribose 5-phosphate. So the ribose 5-phosphate will now become phosphoribosyl pyrophosphate. Will now become phosphoribosyl pyrophosphate. All right, this anyway it is there. Okay, now here the two phosphates that have been taken from the ATP, they go into the alpha configuration on C1 and form two phosphates. This we call it as phosphoribosyl pyrophosphate, or also called as PRPP, phosphoribosyl pyrophosphate or PRPP. Now the enzyme required for this reaction is called as PRPP synthase. It's called as PRPP synthase. Alright. Now this step is regulated. This step is regulated if a inorganic phosphate is there. It is a positive regulator. Inorganic phosphate is a positive regulator, whereas nucleotide monophosphates, nucleotide diphosphates, nucleotide triphosphates, all these are negative regulators of this step. There is one more thing which inhibits this step, that is the 2,3-BPG of Rappaport-Lubering cycle. 2,3-BPG of Rappaport-Lubering cycle. This also inhibits the enzyme PRPP synthase. This you need to remember. It can be a MCQ bit. 2,3-BPG from Rappaport-Lubering cycle inside the RBCs. It also inhibits this step PRPP synthase. Alright, so this is the first committed step. That means ribose 5-phosphate from hexose monophosphate shunt. From there, ribose 5-phosphate has come. This ribose 5-phosphate is now converted to phosphoribosyl pyrophosphate, and ribose 5-phosphate is now committed. It is now committed. So you can also say that this is the committed step. This is the committed step in nucleotide synthesis. But this PRPP, like our glucose 6-phosphate, it has many roles. What can happen to this PRPP? This PRPP, phosphoribosyl pyrophosphate, it can go into purine synthesis. It can go into purine synthesis. It can also go into pyrimidine synthesis. It can also go into pyrimidine synthesis. It can also go into salvage pathways. Also go into salvage pathways. It can also take part in histidine metabolism. Histidine metab. Also it can take part in tryptophan metabolism. Tryptophan metabolism, especially in the formation of NAD by Priest-Handler pathway. Priest-Handler pathway. If you remember our tryptophan metabolism, also we have talked about this in niacin. In both those classes, we have talked about Priest-Handler pathway and Dietrich pathway. Two pathways exist for conversion of tryptophan into NAD or nicotinamide dinucleotide. Okay, so in Priest-Handler pathway, PRPP will take part. So PRPP, once it is formed from ribose 5-phosphate, once ribose 5-phosphate forms PRPP, now it is committed that it has to go somewhere. So it can either go into purine synthesis or pyrimidine synthesis, salvage pathways, histidine metabolism, or tryptophan metabolism. All right. Okay. Now let us look how this PRPP enters this purine synthesis pathway. PRPP, phosphoribosyl pyrophosphate. Now we'll just look into structures. No harm in spending some time to understand few things. Okay, so here we have seen that this is there in alpha configuration 2 PIs. This is there in alpha configuration 2 PIs, and a phosphate group is attached. Phosphate group is attached. Okay, now this is our PRPP or phosphoribosyl pyrophosphate. This phosphoribosyl pyrophosphate, it will take an amino group. It will take an amino group from glutamine. It will take an amino group. If you remember amino acids, glutamine is an amide of glutamate. So what will happen if glutamine loses an amino group? It forms glutamate, and at the same time these two phosphates, they are liberated as PPI. Now this PPI is broken down to 2PI so that this reaction can proceed forwards only, and this step requires an ATP molecule. Basically, this step wherein PRPP takes an amino group from glutamine, this step is totally reversible, but because this PPI is broken down into two inorganic phosphates by ATP, this reaction will go forwards only. So what will happen now is this ribose sugar with C1 okay, so the amino group that has been taken from glutamine, this is attached to C1. This is attached to C1 in a beta configuration. Here it was in the alpha configuration. So the bond or the functional group that is being attached gets converted, or the C1 configuration gets inverted during this step. Okay, so an alpha pyrophosphate which is attached in an alpha configuration to C1 is now having amino group in a beta configuration on the same C1. A glutamine is utilized to form glutamate. Pyrophosphate is converted to two inorganic phosphates. The enzyme required for this reaction is called as PRPP glutamyl amido transferase. PRPP glutamyl amido transferase. From PRPP pyrophosphate is removed, and from glutamine amino group is attached, and this now forms 5-phosphoribosylamine. Forms 5-phosphoribosylamine. All right. So PRPP now gets converted to 5-phosphoribosylamine. Amino group is taken from glutamine, which forms glutamate. PPI becomes 2PI, and this step is called the flux regulating step. Flux regulating step of the entire purine synthesis. All right, this PRPP amido transferase, because this is the first committed step after formation of PRPP or into the purine synthesis, and it is regulated by the product. It is highly regulated. That is why this is also the rate limiting enzyme. This is also the rate limiting enzyme for the whole reaction. So let us go back once more. Ribose 5-phosphate is the substrate. This ribose 5-phosphate, in presence of PRPP synthase, it forms phosphoribosyl pyrophosphate. 2,3-BPG and nucleotides are inhibitors of this, and PI is the activator of this. This PRPP goes into pyrimidine synthesis, purine synthesis, histidine, tryptophan and salvage pathways, and this PRPP gets converted to 5-ribosylamine with the rate limiting step PRPP glutamyl amido transferase. Purine synthesis is something like growing a tree. Suppose on the right hand side I'm drawing a small pot. Okay, so this is the pot in which you are growing your tree. All right, and this tree grows like this. All right, then grows like this, and finally like this. All right, so consider this pot as your PRPP, or say ribose 5-phosphate. Consider this pot as your ribose 5-phosphate on which this purine ring has been synthesized. Okay, now just follow me. Most easiest way, I'll put on the right hand side, I'll draw the how the purine is being structured on ribose 5-phosphate, and on the left side we'll start writing the names. All right, so first is your ribose 5-phosphate. I'll just write this as here ribose 5-phosphate. All right, to this ribose 5-phosphate, let this be the phosphate that is attached, ribose 5-phosphate. Okay, so in the first step, what is happening? PRPP is formed. Okay, in the first step PRPP is formed. This PRPP, because of PRPP glutamyl amidinotransferase, these two are removed as 2-pi. Okay, PRPP glutamyl amidinotransferase, or rate limiting enzyme, is the one which is doing that. So first step is ribose 5-phosphate getting converted to PRPP. In the second step, this PRPP is removed and an amino group is attached. Where is this amino group coming from? Yes, it is coming from glutamine. Okay, it is coming from glutamine. Now after this step, we have seen till here, in presence of PRPP glutamyl amidinotransferase, these 2-pi is removed and glutamine gives an amino group to glutamate, and the first compound that forms is 5-phosphoribosyl amine. 5-phosphoribosyl amine. Okay, this step here, it requires ATP. So wherever ATP is required, I'll put a red dot. Wherever ATP is required, I'll put a red dot. All right, now so in this step, first step, ribose 5-phosphate forms phosphoribosyl pyrophosphate. 1 ATP is required, and from pyrophosphate to inorganic phosphate, another ATP is required. Now 5-phosphoribosyl amine is formed. In the next step, glycine is added. So glycine, you remember glycine to be a simplest amino acid with a carboxylic group, and an amino group is the hydrogen. All right, so in the next step, a glycine is added. This step also requires, this step also requires an ATP molecule. This step also requires an ATP molecule. So this is called as glycinamide ribosyl 5-phosphate. Glycinamide ribosyl 5-phosphate, or GAR. Alright. Now, to this glycinamide ribosyl 5-phosphate, a carboxyl group is attached. A carboxyl group is attached from tetrahydrofolate, that is N5N10 methyl tetrahydrofolate, which gives a carbon and it gets converted to tetrahydrofolate. Okay, so this is the one which is giving this carbon, and this was given by your glycine. It's given by glycine. All right. So this step, N5N10 methyl tetrahydrofolate, this doesn't require any energy. The enzyme for this is formyl transferase. The compound now that is formed is formyl glycinamide ribosyl 5-phosphate, also called as FGAR. Formyl glycinamide ribosyl 5-phosphate. All right. In the next step, what happens is another glutamine molecule, it will add another glutamine molecule, it will add another amino group, this amino group, again it is coming from glutamine. Okay, glutamine gives amino group and forms glutamate, and this step also requires an ATP. Okay, now the compound that is formed is one more amino group is added. So this formyl glycinamide becomes formyl glycinamidine ribosyl 5-phosphate, or FGAM, formyl glycinamidine ribosyl 5-phosphate. All right. Next, in the next reaction, what happens is there is cyclization of this compound, and it forms an imidazole compound. Wherein this carboxyl group is removed and a double bond is formed. Okay. All right. So this compound that is formed, I'll remove the extra hydrogens because we get confused. All right. So this compound that is formed is an imidazole ring, and the compound formed after cyclization reaction, is called as amino imidazole ribose 5-phosphate, or AIR. Amino imidazole ribose 5-phosphate, or AIR, and this requires an ATP, and this requires an ATP. In the next step, a bicarbonate gives a carbonyl group. A bicarbonate gives a carbonyl group. Bicarbonate gives the carbon dioxide, or bicarbonate or carbon dioxide will give this, and this will form amino this will form carboxamide aminoimidazole ribosyl 5-phosphate, or CARE. Aminoimidazole ribosyl 5-phosphate, or CARE. This also requires a ATP. This step also requires an ATP. Alright, in the next step, what is happening is aspartate. In the next step, aspartate gives an amino group. Aspartate gives an amino group and forms fumarate back. Aspartate gives an amino group and forms fumarate back, and this step requires an ATP molecule. This step requires an ATP molecule, and the compound form, there are two compounds formed. The first compound formed during this is succinyl carboxamide aminoimidazole ribosyl 5-phosphate, or SCARE, and the subsequent step when fumarate is liberated is aminoimidazole carboxamidine ribosyl 5-phosphate. Okay, now the final carbon that is attached, it comes from N10 formyl tetrahydrofolate. It comes from N10 formyl tetrahydrofolate, and now it becomes formyl aminoimidazole carboxamidine ribosyl 5-phosphate. Okay, this step doesn't require an ATP. And the final step, that is IMP cyclohexase. IMP cyclohexase will complete this cycle. IMP cyclohexase will complete this cycle, and it will form a complete purine ring, or inosine monophosphate. It will form inosine monophosphate in presence of IMP cyclohexase. Alright, so first what had happened, ribose 5-phosphate had formed PRPP. PRPP had formed 5-phosphoribosyl amine, which has taken a glycine molecule. It has formed glycinamide ribosyl 5-phosphate. Now this has attached a carboxyl group from tetrahydrofolate to form formyl glycinamide ribosyl 5-phosphate. Then an amino group was attached, and it formed formyl glycinamidine ribosyl 5-phosphate. Then after that, carbon dioxide gave this carboxyl group, then aspartate gave this nitrogen group, and finally N10 formyl tetrahydrofolate gave this carbon to form the complete IMP pure. So this is how an IMP is formed. Now let us go back, have a thing like first there was ribose 5-phosphate. This ribose 5-phosphate was converted to PRPP by Prpp synthase. Now this PRPP was converted to 5-phosphoribosylamine in presence of enzyme PRPP glutamyl amido transferase. This is the rate limiting enzyme. Okay, this is the rate limiting enzyme. This 5-phosphoribosylamine is formed when a glutamine gives an amino group. This is the N9 that is formed first. So this N9 is formed from glutamine. After that, glycine is attached. In presence of glycinamide ribosyl 5-phosphate synthase, ATP is utilized and glycine gives the entire molecule. So the entire molecule of glycine is incorporated here. Okay, and this will be the C4, C5 and N7 of the molecule. After that, formyl transferase will give C8. Formyl transferase will give C8 from folic acid. Okay, after that what happens? Another glutamine, the N3 amino group comes from other glutamine. Okay, this step also requires energy. After this step, what happens is a carboxylase or amino-imidazole, before that what happens is there is cyclization reaction which requires ATP and amino-imidazole ribosyl 5-phosphate is formed. An imidazole group containing 2 carbons, 2 nitrogens and the C8, it forms an imidazole group after this cyclization reaction, okay, which requires an ATP. Now after that, AIR carboxylase or amido-imidazole ribosyl 5-phosphate carboxylase will add the C6 carbon from bicarbonate or the carbon dioxide. All right, next N1 is added from aspartate and C2 is added from another folic acid. C2 is added from another folic acid. Finally, IMP cyclohexase, it will complete the ring structure. It will complete the ring structure. So a complete inosine monophosphate is formed. So inosine monophosphate is the first purine nucleoside that is formed. Okay, IMP or inosine monophosphate is the first purine nucleoside that is formed. First ribose 5-phosphate, then PRPP, then ribosyl amine. All right. Now this IMP will be metabolized further, but before that, let us look into some of the inhibitors that are there. Now, what color do I use for the inhibitors? Okay, let's go with dark blue. All right. So the first inhibitor that we have seen here is the 2,3-BPG that you need to remember, doesn't matter. Okay, glutamine, the first glutamine that is added because of our enzyme PRPP glutamyl amido transferase, this is inhibited by diazonorleucin. So at N9 of glutamine, you have an anti-cancer drug diazonorleucin, which inhibits the formation of 5-phosphoribosylamine or the attachment of this N9. Here on this glutamine or N3, we have another inhibitor called as azaserine. We have the inhibitor azaserine which will inhibit this reaction and it will prevent formation of formyl glycine amidine. The first one was inhibiting this reaction. The second one is inhibiting this reaction, formation of formyl glycine amidine ribosyl 5-phosphate. Then wherever we are having folic acid, it is inhibited by sulfonamides. It is inhibited by sulfonamides here also. It is inhibited by sulfonamides. Why? Because they have a structure very similar to PABA, which is a component of folic acid. Okay, so azathioprine, diazoxyl and sulfonamides are the inhibitors that you need to remember at this step. Going further, IMP or inosine monophosphate that has formed, it can either form adenylosuccinate, or it can form xanthine monophosphate, The enzyme required for conversion of IMP to adenylosuccinate or AMPS is adenylosuccinate synthase, whereas the enzyme here is IMP dehydrogenase. Now, talking about energetics, adenylosuccinate synthetase, it requires a GTP, which gets converted to GDP plus PI. Alright, and the amino group that comes on IMP is from aspartate. I hope you remember my part 1 class of nucleotide chemistry, wherein we have seen what is IMP, what is adenine, what is guanine, structure of IMP and all that. So inosine monophosphate, the structure is very simple. It is like this. Alright, now what we are doing is we are attaching a aspartate group here. To this IMP we are attaching an aspartate group. So this IMP with an attached aspartate group is the adenylosuccinate. Alright. Now this adenylosuccinate, it gets converted to adenosine monophosphate or AMP in presence of adenylosuccinase. The attached aspartate gives an amino group to this and it comes out as fumarate. The aspartate is coming out. Remember in the previous slide also, aspartate gives this amino group and comes out as fumarate. Same way here aspartate comes in, it forms adenylosuccinate. This will give the amino group to the IMP and it will come out as fumarate, and the product form is adenosine monophosphate. So how exactly it will look? It will look like this. This amino group has come. Okay, this amino group has come and it has now become adenosine monophosphate. Important thing for you to remember here is this: there is requirement of GTP at this step. Now IMP undergoes dehydrogenation. In presence of IMP dehydrogenase to form xanthine monophosphate. What is xanthine monophosphate? It is nothing but this is your inosine monophosphate, or this is your hypoxanthine with attached ribose and it is IMP. So to this xanthine monophosphate, to this IMP, another reduction step will give a carboxyl group and this becomes your xanthine monophosphate. How this has happened? Because of this dehydrogenation reaction. Now this xanthine monophosphate, it gets converted. This xanthine monophosphate takes an amino group. It takes an amino group. Who is the amino group donor? Yes, it is the glutamine. This glutamine will form glutamate. It will give the amino group. The amino group is attached here. And this will become guanosine. This will become guanosine monophosphate. Okay, so it will become guanosine monophosphate. So this is how we get our GMP and AMP. All right. One thing you need to remember at this step of guanosine is this step will convert ATP to AMP plus PPI. This PPI is further converted to 2PI utilizing an ATP. So there are 2 ATPs involved at this step, whereas here there is one GTP that is involved. Okay, there is one GTP that is involved. Coming to inhibitors of this step, 6-mercaptopurine. 6-mercaptopurine is the inhibitor of adenylosuccinate synthetase and IMP dehydrogenase as well. 6-mercaptopurine is an anti-cancer drug used in ALL and NHL. So it is an inhibitor of adenylosuccinate synthetase as well as IMP dehydrogenase. IMP dehydrogenase is also inhibited by mycophenolic acid, an immunosuppressant, mycophenolic acid. Okay. Now, what will happen to this AMP and GMP? This AMP will get converted to ADP, which will get converted to ATP. This GMP will get converted to GDP, which will get converted to GTP. How they are getting converted? Every time they are utilizing an ATP. Every time they are utilizing an ATP to form ATP. All right. Here we are having kinases, nucleotide monophosphate kinase, nucleoside diphosphate kinase, nucleoside monophosphate kinase, nucleoside diphosphate kinase. These ATP, GTP can get further converted to DATP or DGTP from DGDP, from DADP by an enzyme ribonucleotide reductase, which requires thioredoxin. That is a total different video. It's a big topic. We'll discuss that in the next video perhaps. Okay, for now you remember that IMP has formed ATP and GTP. Let us look at the number of ATPs required. Right from the beginning, from Prpp synthase, how many ATPs were required? Let us count the flowers here. 1, 2, 3, 4, 5, 6, 7. 7 ATPs for formation of, from ribose 5-phosphate to IMP. We have requirement of 7 ATPs. Whereas from IMP to AMP, from IMP to AMP, 1 ATP is required. From IMP to GMP, 2 ATPs are required. Okay, from IMP to GMP, 2 ATPs are required. So total of 9 ATPs for formation of GMP and 8 ATPs for AMP formation. That huge amount of energy. Just remember, this is only for one nucleotide. We require 8 and 9 ATPs. Imagine for the entire DNA, for the cell to replicate, how much energy is required. All right, this